Now, translation. So what we just covered with going from DNA to mRNA is transcription. Translation is different. So what you do for translation is you take RNA and then you finally make that protein. And again, those ribosomes, those enzymes, very important. So what you're doing is creating polypeptides from mRNA. So in this cartoon, each of these little beads is amino acid. Remember, those amino acids are linked via peptide bonds. So when we have many peptide bonds, you have a polypeptide. So the thing is that ribosomes, these are enzymes. Just like RNA polymerase reads DNA to make mRNA, ribosomes read mRNA and translate it into proteins. So what we have here is a ribosome. So what do they do? So they take all these amino acids, and again, at this level, you should just know that there are 20 general amino acids, not including selenocysteine. So amino acids are taken, and actually this is where tRNA comes in handy. So tRNA is actually responsible for ferrying amino acids to a ribosome. And then the ribosome reads a mRNA molecule, makes a polypeptide by putting the linked amino acids together via peptide bonds, and then you finally get a polypeptide. So the overall process is called translation. Now, this is where I don't want you to get too much into weeds. This is just for your information, but this is how you're able to actually get mRNA to be from mRNA to make a protein. So this is what we have here. So this blue blob is a ribosome, and this little, like, how would I describe this? This kind of like, I've heard described like kind of like a cross or a little blob right here, but this is a tRNA, and this is actually amino acid. So notice that these tRNAs are actually carrying the amino acids, and then this ribosome is starting to connect all the amino acids together. That's pretty much what I want you to know. Do I want you to know all this information over down here? This is more FYI. I just want you to know that ribosomes are the ones that read mRNA and they make a polypeptide from it. That's pretty much it. Now the genetic code, you might have seen that, but what does this alphabet soup mean? Well, notice that you see that there are these 4 letters, and DNA you have the 4 letters. So A, T, C, G, and mRNA, you have, instead of T, you have U. But notice that when you go from this here to over here, you notice that you have this kind of base pairing, right? So A and T, A and U. So these follow these base pairings. You just flip it around. And then what you have is that the interesting thing is that how does the ribosome know which amino acid to put at what position? Well, they actually read 3 base pairs, or not base pairs, they read 3 bases of an mRNA molecule at a time, and then and each possible combination of 3 bases corresponds to an amino acid. So again, genetic code, do I want you to know and memorize what triplet means what amino acid? No, I don't think even molecular biology text, because there are 64 potential codons, and I don't think they're not going to test you, or at least when I was an undergrad, they didn't test me on that. You're not going to be tested on that here. But what you should know, the genetic code refers to that whole sequence of these bases, all those letters. So the bases are kind of like the alphabet. So in DNA, you have a 4-letter alphabet. And RNA, you also have a 4-letter alphabet. Mostly the same, but you just change U and T. Yeah, so don't know about initiation, elongation, and termination. If you're studying that, that's beyond the scope of this class. All right, so codons, these are the words of the genetic code. So, and the words are 3 bases, so it's consistent. Ribosomes read 3 bases at a time, so this doesn't really change. So when you have a gene, it's like putting multiple words together in a sentence. So genes are kind of like paragraphs or novels. So just like how you put words together to describe something or to give information on how to make something, same with genes. So by having all these codons together, they basically give you the information to make a new polypeptide. Now translation. So translation, this is where it's often kind of easily confused. So translation, say, or if you're talking about someone about translation in a non-molecular biology context, what do they think of? Well, they might think of translating between languages, right? So what we have here is a translation, same information, but now you're going to a different language. So yeah, I study anatomy and physiology, j'étudie l'anatomie et la physiologie in French. I know I need to do more in Duolingo, but same information, just a different language. So when you're going from mRNA, you're going to a different language with proteins. You still have that same sequence, but now you're translating to a different language. So this is another way I remember it between transcription and translation. With transcription, you're going from DNA, you're going from nucleic acid to nucleic acid. But when you're going from mRNA to protein, you're going from nucleic acids to amino acids. So again, DNA to RNA, nucleic acid to nucleic acid. mRNA to protein, you're going from nucleic acids to amino acids, so a different language altogether. At least that's how I like to think of it. So mRNA, so this is another analogy. mRNA is like a recipe, and the ribosome is the enzyme that takes the information from that recipe, and then it takes all the amino acids, and based on the information in the mRNA, it's going to assemble that protein based on that information. So you might have heard of this if you've taken a biology class, central dogma. So I know it's like kind of an outdated term, but when you're starting out, it helps to convey how information is transferred from DNA to make a protein. So it's like a recipe book, the very old recipe book you don't want to alter or damage even further. Then transcription is how you get that information from the recipe book. So when you're making an mRNA transcript, you're not copying the entire genome and all the 3 billion base pairs in a DNA molecule. Your cells are kind of choosing what information they need to make what proteins they need. So when you have that partial transcript of that original DNA, you have translated, and then you finally get that polypeptide you can use to make the proteins in your body. And how is this very relevant? So this is how if you've had the Pfizer Moderna mRNA vaccines, this is the mRNA we were talking about is the same mRNA that is the core of these vaccines. Now here's that infamous coronavirus, but are they injecting the entire coronavirus into your system? They're actually not. The cool thing about this is that they actually only take the part that encodes those proteins. So they only take a tiny part of the sequence. They don't take the whole information of a virus. They only take the part that encodes these spike proteins. So spike proteins, what they do is they only take the small part, the one that has these spike proteins on the outside. And this is how they do the vaccination. They have all these, like, remember those phospholipid membranes, and so it's kind of like they have an artificial membrane. So what can this membrane do? It can actually help to deliver and carry these mRNA or mRNA molecules into your cells, because again, these mRNA molecules by themselves, they would actually have a hard time. So by making it kind of like a vesicle, it's almost like endocytosis, that moving of or shuttling from something from the inside, or actually not quite endocytosis because it's not actually pinching. I retract that part. But somehow this is how you're able to actually get that mRNA inside your cells. So what happens is that this mRNA goes to a ribosome. That ribosome translates it and makes a spike protein. And this spike protein is how it's processed by your cell even further, and then it presents it to immune cells. So this is how these vaccines work. And you might have heard, like, so again, translation or transcription to translation. So DNA to mRNA protein. The cool thing about the Pfizer and Moderna vaccines is that they kind of take a shortcut. They don't inject the entire viral genome because you don't want that virus to actually replicate in your body. They only take the part that encodes that spike protein, and then when you deliver that mRNA into your cells make the spike protein, and then they can present it to your immune system, so your body is able to recognize the spike proteins of a coronavirus before it's actually infected with a coronavirus. So again, common misconception is like they're injecting the entire virus with these vaccines. Nope, they're only using this part, and the spike protein. The spike proteins are only on the outside. They don't actually contain all the enzymes and information the coronaviruses actually need to replicate. So it's like if you just took someone's hand and then just like try to make a new person from that person's hand only. It doesn't have the reproductive organs, and that's the same thing with the spike protein RNA in these vaccines. They're only trying to use a part that your cells, your immune cells can recognize, so it can actually combat the full virus. And another thing you may have heard, misconception you may have heard, is like these mRNA vaccines are altering your DNA. If you look at the flow of information, mRNA doesn't flow back to DNA. mRNA does not alter DNA. So again, this is where a lot of misinformation is happening. That flows from RNA to protein. It doesn't flow the other way around. So Pfizer and Moderna do not alter your DNA.